Hi everybody. Now we are going to watch about downstream processing. Introduction. The extraction and purification of biological product from the fermentation broth is called downstream processing or product recovery. Downstream processing is quite complex and variable depending upon the type of the product. Broadly, it can be divided into 5 stages: cell harvesting, cell lysis, breakage of the cell, concentration, purification, and formulation. First, we will see about cell harvesting. Once the fermentation is complete, the solid phase is separated from the liquid phase by any of the following methods like flotation, flocculation, filtration, and centrifugation. First method is the flotation method. A gas is passed through the fermentation broth. This gas forms tiny bubbles to which cells get absorbed. These bubbles rise to the surface of the broth and form a foam. Sometimes, along with the gas, collector substances are added which facilitate foam fermentation. The foam contains the cell is removed. Next method is the flocculation. At high cell density, some cells, like yeast cell, aggregate and thus settle down at the bottom of the fermenter. This process can be accelerated by the addition of flocculating agent like salts, organic polyelectrolyte, and mineral hydrocolloid. And next is the filtration method. It is the most common type of cell separation technique. Many types of filters are being used. One is the depth filter. These are composed of filamentous material, for example, glass wool, asbestos, or cellulose paper. Cells get entrapped in the matrix and clear cell-free material is passed out. This technique is used for the separation of filamentous fungi. Asbestos filters. These filters have specific porosity which can retain cells and let go liquid material. This is most suited for separation of bacteria. Rotary drum vacuum filters. This is usually applied for the separation of yeast cell. This unit consists of a rotary drum which is partially immersed in a fermentation broth. Rotating drum absorbs cellular biomass. Once the biomass is out of the fermentation broth, it is partially dried and then removed from the drum. Centrifugation. This is a process of separating cells from the liquid based on the difference in their density. The cell culture is spun at a high speed in an instrument called centrifuge. This results in settling of denser material, that is, cells at the bottom or sides of the holder. In industry, continuous flow centrifuges are used in which culture is continuously supplied to the centrifuge. The cells get collected at the centrifuge and cell-free supernatant flows out of the centrifuge. Next step is the lysis or breakage of cells. If the desired product is located inside the cell, then the cells are first recovered from the fermenter. These cells are suspended in buffer and their cell walls are disintegrated so as to break open the cells by physical or chemical means in order to release intracellular products in the medium. Physical method includes ultrasonication. The cells are disrupted by passing ultra waves through samples. This technique is ideal in laboratory where sample size is small. Next is the osmotic shock. The cells are suspended in viscous solutions like 20% sucrose or glucose. The cell suspension is then transferred to the cold water, which results in cell lysis. Next method is this heat shock, thermolysis. The cells are exposed to heat, which results in disintegration of the cell. It is an economical method, but the product has to be heat stable. Next is the high pressure homogenization method. The cell suspension is forced to pass through a narrow pore at a high pressure, which results in breakage of cells. Grinding with glass beads. The cell suspension containing glass bead is subjected to a very high speed in a vessel. The cells break as they are forced against the walls of the vessel by the beads. Next is the chemical method. Various chemicals can destroy the cell walls, cell membranes of the cell. These chemicals can be alkali, that is sodium hydroxide, and solvents like alcohols and detergents like sodium lauryl sulfate, triton X, and red cetyl trimethyl ammonium bromide. Enzymatic lysis. The cells are broken with the help of enzymes. Bacterial cells are lysed by the addition of lysozyme. This enzyme hydrolyzes beta 1,4 glycosidic linkage of peptidoglycan layer of the bacterial cell wall. Fungal cells are lysed by the addition of chitinase, cellulase, and mannase. Next step is the concentration. More than 90% of the cell-free supernatant is water, and the amount of desired product is very less. Evaporation process concentrates the requisite product. Water is evaporated by applying heat to the supernatant with or without vacuum. The amount of heat applied is decided by the heat stability of the product. Various forms of evaporators are used. For example, plate evaporator, falling film evaporator, forced film evaporator, and centrifugal forced film evaporator. Next is the liquid-liquid extraction. A desired product can be concentrated by the transfer of solute from one liquid to another liquid. This process also results in partial purification of the product. The liquid extraction is basically of two types: extraction of low molecular weight product, extraction of high molecular weight products. Extraction of low molecular weight product. This is primarily applied for extraction of lipid soluble compounds. This process can be carried by the following technique: physical extraction. The aqueous solution containing the product is mixed with an organic solvent. The amount of product getting into organic solvent depends on the partial coefficient of the product. Reactive extraction. It is like physical extraction procedure, except that the product is first reacted with a carrier compound and then mixed with an organic solvent. Extraction of high molecular weight. This technique is applied to compounds like protein, which get inactivated in the presence of organic solvents like ethanol, methanol, chloroform, hexane. These compounds are extracted by using an aqueous two-phase system. In this technique, a polymer, example polyethylene glycol, and a salt solution, example ammonium sulfate, are added to the aqueous solution containing the product to be concentrated. The particulate matter, that is, cells, remain in one phase and the proteins are transferred to another phase. The solubility of the desired product is dependent on various properties of the protein, for example, ionic character, hydrophobicity, and size. Next is membrane filtration. This technique involves the use of semi-permeable membrane. Many kinds of membranes are available based on the size of pores and their chemical nature. Polyether sulfane or polyvinyl difluoride. Unfortunately, these filters cannot be sterilized. Now membranes made up of ceramics and steel are being introduced, which can be easily autoclaved. Membrane filtration techniques like macrofiltration, ultrafiltration, and hyperfiltration. Membrane adsorber. The membrane contains charge groups or ligands to which a desired product can combine specifically once the aqueous solvent containing the product is passed through this. The adsorbed material is then eluted using various buffers and salts. Next step is the precipitation. This is the most commonly used procedure for concentration of compounds, especially proteins and polysaccharides. The agents commonly used in the process of precipitation are neutral salts like ammonium sulfate, organic solvents like ethanol, acetone, propanol, and non-ionic polymers, that is PEG, polyethylene glycol, and ionic polymers like polyacrylic acid and polyethylene amine. Next step is the purification by using chromatography. Chromatography is a procedure for separating molecules based on their size, charge, hydrophobicity, and specific binding to ligands. The chromatography techniques used are as follows: gel filtration chromatography, which is also known as size exclusive chromatography. The matrix is made up of tiny beads having many pores in them. Many types of beads are available having different porosity. Some molecules enter the beads, whereas large molecules cannot enter and therefore come out of the first column first. By this technique, protein of variable size can be purified. Next method is the ion exchange chromatography. Most of the proteins have a net positive or negative charge. This property of the protein is exploited for the purification of proteins by passing protein solution through columns of charged resins. Two types of resins are used in the industry. One is cation exchange resin and anion exchange resin. Cation exchanges like carboxymethyl cellulose have negative charged groups, whereas anion exchanges like diethyl amino ethyl have positive charged groups. Protein carrying net positive charge bind to cation exchanges, whereas proteins carrying net negative charge bind to anion exchanges. Next method is the affinity chromatography. In this procedure, the proteins are separated based on their affinity for a product compound, that is ligand. Once the protein is bound to the affinity matrix, it is eluted by changing the pH of the eluting buffer or alteration of the ionic strength. Thank you for watching the video. Have a great day.